Hello, good afternoon. We have a lot of things to do today. We need to finish lecture 6, and then we're going to start a little bit of lecture 7 as well. So thank you for being on time, but we're going to get started right away. So last class, we learned about the process of DNA replication, which is something that needs to happen right before a cell divides. Okay? And then we talk about the purpose of why cells need to divide. They divide because of growth and development, um, or perhaps we need to replace lost or damaged cells. And for some organisms, cell division is a way of asexual reproduction. Okay. So today we're going to start by uh, talking about the cell cycle. The cell cycle. Right. So let's draw this thing together. Here is a pie chart basically, and we're gonna break this down into into two main events here. Right? So this bigger portion of the pie. We have a name for that. I'm gonna just draw it right here. This is called interface. Interface. Right? All the way over there. So let's draw this out first, and then I'm going to tell you more information about it. Okay. No, we're just getting started. Okay. For those of you who just joined, we are just starting with a note here. All right. So this interface is where the cell spends about 90% of its time in this phase, right? And then the 10% of the time is going to be spent doing the actual cell division, which we call mitosis. Okay? So about 10% of its time. Now, of course, these are average number. Some cells spend longer in interface, some cells spend shorter in interface, but roughly speaking, it's about 90% of its time. Okay. Some cells actually don't divide either, right? Like if you think of heart cells, they don't divide, so they would spend their entire cell life within this interface. So, what exactly is happening within the interface? Well, we can actually divide it into further sub stages, if you would. Okay. So here, this is called the G1, this is called the S phase, and this is called the G2. So during G1, what the cell is doing, it's basically performing its normal function. Okay, normal functions, whatever it's supposed to do. Right? If it's a liver cell, it's going to be filtering blood, perhaps. If it's muscle cell, then it's going to be contracting. Whatever its normal function it is, it's going to be doing it in G1. And this would represent a cell that just finished dividing. And so at that point, it's pretty small, right? It's not ready to go through another round of division. And so in G1, it's basically going to be focusing on growth. It's going to get bigger and bigger over time. And then eventually, when it is big enough, then the cell is going to consider division again, right? Consider to go through another round of splitting. But before you do that, you need to copy all your genetic material, right? And so that's what the S phase is. The S phase is for DNA replication. S is for synthesis, DNA synthesis, DNA replication. So you're going to copy all your genetic material. And then in G2, you're going to be grow some more. Grow some more. And then final preparation for division. Okay. This is it. This is the final stage. You gotta make sure that you have all the resources that you need to finally do the division. Okay. All the while, the cell is it's still be able to be performing its normal function. Okay. Now over here, we have to add what we call a G1 checkpoint. Okay. These checkpoints are important because before you allow the cell to copy its DNA, there is like a checklist that you have to go through to make sure the cell is ready for this next stage. For example, you need to check the DNA quality. Right? You want to check for mistakes, damages. Right? Like if you find base pair that are mismatched, then you're going to want to repair it before you make a copy. If you find DNA damage, you also want to fix that before you do the DNA replication. What are some of the other things that you should check for before you allow the cell to copy its DNA? Just like throw me some ideas, guys.
Maybe the condition of I forget what they're called. The what's on the end of the
Yeah,
chromosome.
exactly. Very good, right? So the telomeres that they're called. So telomeres, telomere length, right? Like, is it too short, right? If it's too short, it's not a good idea to copy the DNA because you're gonna start cutting into critical DNA, right? We also want to check for the health of the cell. Like, is it healthy? Is it big enough? Right? Big enough. Any damage to the cell, right? Enough resources. You have age of the cell, sure. Enough resources, right? What kind of resources are we talking about? Like, does it have enough ATP? Does it have enough nucleotide? Right? That's what you need to make DNA. You need ACTGs and you need energy to do it. So that's like a checklist to go through. Your cell's gonna do this check. And then if all is good, then it gives the green light to go ahead and copy the DNA. Okay? I'm just taking a pause so you can catch up copying. So of course you're wondering, I assume, what's gonna happen if we don't pass the checklist? Right? What if some of these things are, you know, uh uh not like not pass. I can't think of a better word. Okay. So like the telomeres are too short. So what do you do, right? What if the cell is not big enough? What if there are, not enough resources, right? Well, I mean, it depends on the situation. First of all, if the telomeres are too short, then that's it. There's nothing you can do about it. Then we definitely cannot copy the DNA. So then you pop out of the cell cycle and you enter a growth arrest. Growth arrest. Okay, that means you're not allowed to grow anymore. Right? No more division for you. You can continue to perform the normal cell function. Okay, continue to be a muscle cell. You just can't divide anymore. Okay. DNA quality, if there are mistakes and then damages, you try to repair them. If you're able to repair them, no problem. You can still go on and do the DNA replication. But if the damage are irreparable, if there's nothing else you can do to fix the problem, then you don't really want to keep a cell around that has DNA damage in it. Okay, so growth arrest is not an option in that case. You're gonna probably want to kill it. So that would be apoptosis, programmed cell death, right? Okay. So these are some of the options um, that are available when G1 checkpoint fails. Okay. And sometimes you can just like, wait and grow some more. Right. So some more is two words, sorry. Grow some more. If it's too small, if there is not enough resources, then you just gotta wait. Okay, you try again in a couple days. Okay, maybe it's big enough then, and then you can go through the cell cycle. But that's basically what happens to a cell. Okay, depending on which category failed, if you would. All right, and then it's gonna go and do the DNA replication. This is what we learned last week, right? With the helicase, with the gyrase, and DNA polymerase 3 and 1, and all those glory details. That's going to be happening within the DNA replication stage. And then afterwards, we are going to have another checkpoint. And we're very safe. We got to check before and we're going to check after. This is G2 checkpoint. It's like getting the driver's license in Ontario. To get your license, you got to pass your G1 and your G2. So now the G2 checkpoint, what are we checking for? Well, mostly we're checking for DNA quality again. Right? Did you copy everything? Did you make any mistakes? Any DNA damage? Right? Same thing. Everything got copied. Okay. And if it's good, then again, green light, go ahead, make the final preparation, and then you're gonna get to the mitosis, the actual division. All right. If you have mistakes, then you try to repair it. And if it cannot be repaired, then it goes here. So this is if G2 fails, right? This is if G1 fails. No, there's no S checkpoint, right? Like you basically check before going into S, and then you check when you are leaving S, right, so to speak. So you check before and after S, and you just call them G1 and G2 checkpoint. That's it for the cell cycle. Okay. So it's an important balancing act between when to let the cell go through the cell cycle and when to let the cell to, slow down and stop. If you let the cell progress through the cell cycle, then it's going to divide. Okay. You don't want it to go through too fast because then now you have uncontrolled division, right? But you also don't want it to be too slow because then it's going to affect growth and development and perhaps wound healing at the same time. So it's like a balance between red light and green light. I'm going to use this red light, green light thing a lot for the rest of the class when we explain other concepts related to the cell cycle. Fahima, do you have a question? I see that you're on mute.
When G2 fails, then it will go to G1.
I'm sorry, one more time, please.
When G2 fails, then it will go to G1.
Oh no, when G2 fails, it will go to growth arrest, apoptosis, um,
Oh,
yeah, these
and
kind of
okay,
stuff. Yeah.
okay.
Okay. It's not gonna wait and grow some more because by that time it's like probably too late. So it's more like apoptosis and growth arrest in this case. Okay. And you get the idea. All right. What is next? Let me see. I totally forgot what comes next. Let me just take a look at the slides we had. Okay, yeah. Let's quickly talk about something else first, and then we're gonna go and discuss the details of mitosis. But before mitosis, I want to teach you a few new terminologies. All right, so this is DNA replication. This is lecture 6, okay? Lecture 6. So, we have inherit half of our chromosome from our mom, and half of the chromosome from dad, right? So here is the mother copy of chromosome 1, maternal copy of chromosome 1, paternal copy of chromosome 1, and then maternal copy of 2, and paternal copy of 2. Okay? If you can't hear me, I'm saying maternal, which is like from the mom, and paternal, okay? Which is like from the father. So this is chromosome 1 and chromosome 1, chromosome 2, and chromosome 2. And of course, you have 3, 3, 4, 4, all the way up to 23, 23, right? So these two chromosomes, chromosome 1 from mom and chromosome 1 from dad, they are called homologous. Homologous chromosome. In chemistry, you probably learn about homogeneous mixture, right? So the prefix here means the same, right? Homogeneous like appears to be same, just one phase. Homologous means they are the same thing. Right? So these chromosomes are same length, same size, and they contain same type of genetic information in the same order. Let me write it down for you. Same size, same length, contains genetic info. Sorry, contains same type of genetic information in the same order. Just copy it down first. I'll explain. Okay, homologous chromosome. These ones are also homologous. Homologous chromosome. As an analogy, it's like, imagine you have 23 pairs of shoes, right? Nobody really have that many pair of shoes, but let's say you do have 23 pair of shoes, and you just randomly dump them onto the floor. Right? You would have no trouble finding the missing pair, right? Yeah, like if you get the Nike running shoe, the right one, you're gonna be able to find the left one and then put it together as a pair. Right? If you see a pair of like Crocs, if you see one side of the Crocs, you're gonna be able to find the missing half of the Crocs, right? You can pair them up, right? Why? Because they are same size, right? And they look similar enough. Right? Yet they are not identical. The left shoe and the right shoe are not identical. Like you would know if you try to fit your right foot into the left shoe, right? It's not gonna fit, right? So they're not exactly the same, and that's kind of the idea of what homologous chromosomes are. Right? They are same size, same length, and the genetic information are of the same type. So what do I mean by same type? Like uh, uh, um, of genetic information. Well, let's say over here you inherit like the brown eye gene from your mother because your mom has brown eyes, okay? And then for your dad who happens to have blue eyes, then you would inherit the blue eye gene. So these are both same type of genetic information, eye color, but then it's not the exact same version of it, right? So brown eye and blue eyes are like variation of that genetic information. Okay. But if you find eye color right here, you would expect to find eye color at the same spot on the other chromosome. But whether it is the same gene or not, it depends who your parents are. Right? So another example is like you might find like curly hair here, right? And then you might also have curly hair from the father side. Maybe both your parents have curly hair, then you're gonna inherit curly hair from both of them. Okay. Like I'm oversimplifying things here. We're gonna learn this properly when we get to lecture 8. but for now, this is what you have to know. Okay, maybe further down you have like inherited dimples from your mom side, and then like no dimples from your dad, right? Dimples is like, when you smile, like you have a thing here, right? That's called a dimple, right? But only like like a little dent, right? Okay, so that's genetic apparently. Are we together on this? Any questions? All right, so just humor me here. How many chromosomes do you see on the screen? It's not a trick question, okay? Like you don't even have to answer me. You just answer in your own head, okay? I hope you see 4, right? 1, 2, 3, 4. Right? Obviously, there are 46 in real life, but I'm only drawing 4 here to make it simple. Okay, let's do DNA replication. DNA replication. Okay, so again, maternal means the red copy, paternal means the father copy. Right? Sometimes it sounds the same when I say it. So after DNA replication, you're gonna make a copy of each chromosome. So this chromosome 1 from mom, now you're gonna have 2 of them. We have 2 of them. Okay, they're gonna be stuck together. You're gonna have 2 of number 2 as well. And we're gonna do the same thing with the paternal copy. There we go. Right. So these are identical copies that are going to be stuck together at this location. Okay. The location where the 2 copies are stuck together, it's called the centromere. Don't confuse centromere with telomeres. Telomeres are found at the end of the chromosome. Okay, so here's another centromere, centromere, centromere. All right, I'll give you a definition in a second. Now this copy and this copy are exact copy of each other. They are identical in every way. All the ACTGs are the same. And we call them sister chromatids. Right? This one and this one also sister chromatids. Okay. The red and the blue, still homologous. Okay? But the ones that are stuck together, they are sister chromatids. Okay, so centromeres, that's the location where chromatids, sister chromatids, are attached together. And sister chromatids are identical copies of each other. Okay. So just some new terminologies here. Now, when you're ready, can you tell me how many chromosomes here? Right, over here, 4 chromosomes. How many chromosomes over here? Okay, I see some answer in the chat. Anyone else? Just give me something. Give me your gut feeling answer. Yeah. Yeah. A lot of people would say 8, right? Like I thought it was 8 too. But actually, the people who came up with these rules, I guess, they're like, if it's stuck together, you still count it as 1. Right? So this is 1, 2, 3, 4. So 4 chromosomes. Okay? As long as they're together, you count it as 1. Right? It's like those Kit Kat chocolate bar, right? My kids got a lot of those on Halloween. Right? Together, it's 1. You break it, now you have 2. Now you can share. Right? So still 4 chromosomes, even after DNA replication. Okay. So a couple of new words here that might be confusing for some people. Okay, so the centromere, the chromatids, homologous chromosome, And I just want to help you refresh your memory, I guess. Okay, so like if you take DNA and then you wrapped it around proteins, okay, like a yo-yo. I keep on saying yo-yo, right? Okay. Right. Then this is chromatin. Please do not confuse chromatin, T-I-N, with chromatid, T-I-D. Okay, they sound the same almost, okay? Like it's very difficult to discern. But these are DNA wrapped around proteins. Okay, these red balls, they are proteins. Okay, so chromatin is basically DNA plus protein in the nucleus. It is relatively uncondensed. It's more condensed than naked DNA, like DNA by themselves, but it's relatively uncondensed compared to chromosome. And then it's barely visible under microscope. You can't really see them. And then the chromatin can condense further. Okay, just like really twist themselves out. We did this before. I don't know if you remember. It really twist themselves out, really package around even more protein. Then you get your chromosome. It's like very condensed, visible under the microscope. And the truth is, by the time it gets to the chromosome state, it would have copied itself already. Okay. So then you would get this X shape. And if you have been following what I'm saying, which I hope you have, these are identical copies, so we call them sister chromatids. Right? And then this place where they're stuck together, centromere. Okay, so that's like all the confusing C words that you learned from me today. Chromatin, chromatids, centromere, and chromosome. Any question? Anything you want me to clarify? Okay, so I'm gonna let you copy that. Give me a moment. I'm gonna go grab my jacket. It's like super cold in the basement. 1 second, guys. All right, sorry about that. So that's that. Uh, we'll come back to the note later. We still have quite a bit to write, but for now, let's go back to the PowerPoint slides. Okay, so these are the checkpoint stuff that I told you about. You can read about it yourself. So now we're gonna look at actual cells that are going through the mitosis process. Okay, but first, here is some more note about interphase. Like I said, the cell spends most of its life, about 90% of its time, at this stage. Okay, so once it divides, then it completes the cell cycle, and then we start counting it as like a new life, right? Yeah. Performs regular function, and near the end of this phase, the cell prepares to undergo mitosis, and then the DNA duplicates itself, right? So here is an actual picture of a cell that is an interphase, and you can actually stain different parts of the cell with different colors. right? Using various techniques. And I just want you to know what you're looking at. So the red is basically the cytoplasm, and the blue is the DNA, basically, which of course is in the nucleus right now. And we have the greenish yellow stuff. Um, that's the spindle fibers. And if you don't remember what the spindle fibers are, we learned that in lecture 3. They are microtubules that will guide chromosome movement during mitosis. Mitosis is the actual division, okay? No, I'm not gonna test you on these colors, but then you should know what you're looking at, right? So I'm just writing it out to help you understand what is it that you're looking at. So if you focus on the DNA portion right now, do you see the DNA? Like, I mean, not really, right? Okay. I don't really see anything here, which is expected because at this point, the genetic material exists in the form of chromatin. So the note I wrote for you, chromatin, which is like the natural state of the DNA in the nucleus when the cell is not dividing, it's barely visible under the microscope. Okay, and they still soaked up the dye, the DNA dye. It shows up as a sea of blue, but you can't really make out the individual chromosome. Okay? An example of an analogy would be like think of people at a concert or something, okay? Outdoor concert, tons of people. And then, you're looking at the crowd from a helicopter from above. Right? You know there are people there, but you can't really make out the individual faces and things like that. That's kind of like that. Okay, all this DNA is cramped up in here, but I can't see the individual chromosomes because they're not chromosome yet; they are chromatin. So mitosis, it's separated into 4 phases: uh, prophase, metaphase, anaphase, and telophase. We call this. I mean, there's an acronym you can use to remember it. It's PMET. Okay, PMAT. There's a question. So the chromatin is the outside of the no chromatin and chromosome are same thing. It's just different level of compactness. Okay, so let's take a look at early prophase. Early prophase. So at early prophase, the chromatin coils into visible chromosome. Now, if you look at the blue stuff, you can see the outlines of the chromosome, right? They look like little gummy worms, okay, or like really fat noodles. Right? You can see the outlines now because it's more condensed at this point. By the time the cell goes into prophase, it would have finished DNA replication. And remember at interphase, it was like G1, S, and then G2, and then you go into prophase of mitosis. So if you're at mitosis, you copy all the genetic material already. So at this point, the sister chromatids are held together at the centromere. And just to show you, that's the centromere, and then you can see the 2 sister chromatids. All right. The nuclear envelope starts to disappear so that the chromosomes can come out of the nucleus and get moved around. And the organelle that makes spindle fibers, they are called centrioles, which is something that we also learned in lecture 3. As prophase continues, you can see the nuclear envelope is completely dissolved now, and all these chromosomes are spreading out, so they are even more visible than before. Right? Before they were all kind of cramped up in the nucleus, but now the nuclear envelope is gone. they are able to come out, right? So you see there's chromosome here, and the spindle fibers that are produced by the centromere, so centrioles rather, they will attach to the centromere. Okay, so if you have to draw it out yourself to understand that, that's a chromosome right here with the two sister chromatids, and then you have the centriole here on the ends. I'll label it for you, centriole. And then they will send these spindle fibers and attach to the centromere. Okay, like you might have another chromosome here, and then there will be another spindle fibers attached to it, so on and so forth. Okay, so spindle fibers attach onto each chromosome at the centromere. Okay, so that's it for the prophase. Right? To help you remember what's going on, I guess P is for prepare. You are preparing to split the cell. I know you did some preparation in interface already, but like you prepare more. After P is metaphase, okay, metaphase. M is middle. So there is going to be a tug of war between the spindle fibers on either side of the chromosome, and it's going to get pulled one way and then the other way, back and forth, until eventually they all lined up in the center. Okay, lined up in the middle of the cell. It's pretty much what it says here. So how many chromosomes do you see right here in this picture, in this cartoon? Yeah, 4, right? They're stuck together, so we count it as 1. So 1, 2, 3, 4 chromosomes here. That's metaphase. It's all about lining them up. And then anaphase, A for apart. This is where the spindle fibers pull so hard that you split the sister chromatids. I know they say daughter chromosome here. We're just gonna say sister chromatids. It's, the thing about these things is there's so many different ways to call the same thing, and I wish they were more consistent. But this is sister chromatids. Okay, I probably should change the label in the future. And now, how many chromosomes do you see? How many chromosomes do you see? 1, 2, 3, 4, 5, 6, 7, 8. Right? Okay, so that's it. In metaphase is 4. Once you split it, it's 8. So if the cell divides, if I split the cell in half now, then each half will get 4, right? Each cell will get 4, which is same as the original cell. Right? So the sister chromatid separates, and then they get pulled towards the opposite end of the cell. If you understand everything up to this point, the hardest part is over for mitosis. Because in telophase, right, T for tearing. Okay, you're tearing the cell apart, and then everything that happens here is just the backwards of prophase. Imagine this is like a big blob of dough or Play-Doh. Okay, you tie a rope around the middle like this, and then you pull on the rope. What's going to happen is you will basically pinch the dough like that, and then eventually it will get cut in half. Right? This is telophase, and the actual division of the cell, the cytoplasm getting separated into 2 equal halves, that's called cytokinesis. Cytokinesis. Okay, so everything is reversed compared to prophase. The chromosome is gonna go back to chromatin now, and then the nuclear envelope starts to form again. You need to wrap the genetic material back into the nucleus. We no longer need the spindle fibers. And finally, you just split the cytoplasm. Right? That's called cytokinesis. You gotta make sure that each cell gets roughly the same amount of cytoplasm. Right? They're gonna get equal sharing of the organelles. And, you get half the mitochondria, you get the other half. Right? You get some Golgi, you get some Golgi, like equal sharing, like as much as possible. And that's it. That's how a cell divides. Okay. Let's take a look at this picture. I'm gonna launch a poll Which picture do you think shows anaphase? Which picture? A, B, C, D. Which one shows anaphase?
I have a question.
Yeah, sure.
So, when the cell eventually splits, like you were talking about, for example, the mitochondria. So if the cell originally had like two of them, when it split, the cell would have like one each, or
Yeah,
what,
sure,
or
right? But
like,
mhm. Sorry,
or
go ahead.
like, what did the DNA like the information from the DNA would like make it have two after it split? Do you know what I mean? I don't know if my
Yeah,
question.
yeah. So like, good question, right? But the thing is like mitochondria usually there are like hundreds of them in a cell, right? So like it's roughly 50-50 split, okay? Right? Maybe one will get I don't know, like 45, the other might get 55. Like it's not necessarily like right down the middle 50-50 necessarily, but it's roughly half and half. Okay?
Right. So it's mainly focusing on like the DNA, not really the organelles.
Yeah, so the DNA definitely half-half because the anaphase ensures that, right, when it separates cis chromatids. But then when it comes to the organelles, it's roughly 50-50, but not necessarily exactly 50-50. Does that make sense?
Yes, thank you.
Yeah. Okay, not too many answers. I don't know why the polls are not really popular. But whatever, it is what it is. Most people got this one. Yes, this is anaphase. This is anaphase. Yeah, you can see the individual sister chromatids here. These are onion cells, by the way. Do you see another anaphase? Like which row do you see it, if any? Another anaphase. Anybody? Yep, there's another one right here. That's also anaphase. And Cheyenne said another one right here. Yeah, that's another anaphase. Good. How about this one? What is A? So like I might give you some of these pictures to identify on the quiz and test, right? This one, hopefully you know it's metaphase. Metaphase is right in the middle. Um, this one is telophase. Right? Some people have a hard time telling anaphase and telophase apart. Telophase, you can usually see that the membrane is starting to form in the middle, like where the red line is if you focus. Okay, and also the chromosomes are more concentrated at the poles. Okay, whereas anaphase they are still kind of spread out. You can still make out the individual chromosomes, but here is more uh concentrated because the nuclear envelope is starting to form. You're trying to put them back into the nucleus, right? Yeah, yeah, tighter together, exactly. Um, and this one here is just prophase. Okay. Something like that. It's not mitosis, so it's just interphase. Anybody have any additional questions? All right, here's some concept track questions. Let's just do a poll. It's like not like I said, there's not a lot of participation, so I'm just gonna keep it under 30 seconds, okay? And then I'm gonna move on. So, choose something if you want. Uh, Otherwise, we're just gonna move on. Okay, good. The answer is prophase. Prophase. Let's try another one here. All right, so there are 8 chromosomes at metaphase. How many chromosomes will it have during anaphase? So in metaphase, 8. So 1, 2, 3, 4, 5, 6, 7, 8. And then next is anaphase, which means these things will get split. Right? It'll get split into 2. Oops. Right. Next one gonna get split, 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 split. So it's 8 times 2, which is 16. Okay. This is a question for you to think about. We don't have time to take it up. Feel free to email me what you think if you want. Yeah. Otherwise, I'm sure it's something you can look up on the internet as well. Okay. Next, we're gonna talk about um, those checkpoints, right? The G1 and the G2 checkpoints. So what exactly is performing the checkpoints, the checklist, right? Going through the checklist and making sure everything is okay. Well, actually, these checkpoints are regulated by proteins, okay? And proteins are coded by DNA, right? So if DNA mutates, then the protein will malfunction. Okay? So DNA mutation means your DNA changes, and that could happen because of mistake you made during DNA replication. It could be because of exposure to various things that causes DNA to change, like UV light, that could cause your DNA to change, right? So if the DNA changes, then the instruction to make these checkpoint proteins are going to have mistakes in them. And so the protein might end up not functioning properly. Okay. So there are 2 types of regulators, 2 types of these checkpoint proteins. There are those that are called growth factors, and as the name implies, they are there to promote mitosis. Okay. You can think of them as green lights. On the other hand, you have growth inhibitors, which inhibit mitosis. So we call them red lights. Okay, so when the green lights turn on, the cell is allowed to go through cell cycle and do cell division. When the red lights goes on, then you're not allowed to divide. Right? You pause the cell cycle, you inhibit mitosis. So like I said, you need to have the right balance. You can't have the green lights always on because then the cell would just keep on dividing and dividing and dividing. You can't always have the red lights on either because then nothing would divide. Right? There will be no growth, no wound healing, so on and so forth. There's a question. So when telomeres is too short, it's inhibited by a growth inhibitor. Yes. So to phrase it properly, there would be a protein that would check the telomere length. And if it's too short, then that protein will inhibit mitosis. So in this scenario, the protein that's checking the telomere length, that's a growth inhibitor. Okay. So imagine all these, little proteins that would light up, okay, either as green or as red. And whenever you light up the green one, then the cell divides. And whenever you light up the red one, then it does not divide. Obviously, this is an oversimplification of the actual situation, but it's enough for what we are learning. All right, so I'm gonna give you a quick note here to explain, relatively complicated concept. Um, so please let me know if I'm not making sense. Regulation of cell cycle. Okay. Okay, so we have two kinds of things here. All right, I'm just gonna use red and green here. So we have the growth promoter. Sorry, I call them growth factors. Same thing. Growth factors. And here we have the growth inhibitors. All right, so let's start with a function, which is pretty self-explanatory. Growth factors is going to promote cell division. And on the other hand, growth inhibitor is going to inhibit cell division. Sometimes is like, you're not allowed to divide ever again, okay, because your telomeres are critically short. Sometimes is you're not allowed to divide yet, okay, but if you fix the mistakes, if you come back, then, maybe you can divide again. Okay. Now, as I said, these are proteins, and to make proteins, you need the instruction from the DNA, right? So that's what a gene is. If you forgot, a gene is basically a section, a segment of DNA that has the instruction to make a protein. Every single protein in your body needs to have a gene that tells you how to make them. So the gene to make growth factors, they are called proto-oncogene. Proto-oncogene. Whereas the genes that makes growth inhibitors, they are called tumor suppressor gene. Okay, so normally, on the chromosome, the father and the mother copy, you're gonna have two of these genes. Okay, we have two sets of every gene. So you have a proto-oncogene here and a proto-oncogene here. Same thing for the tumor suppressor gene. Okay. So in other words, you are going to be able to make a pair of green lights, if you would. Okay, so you're gonna have like green lights and green lights. And then over here, you're gonna be making a pair of red lights. Okay. It's a little bit different from the traffic light we are used to, where the green and the red is like on the same thing. Now here we just either making it just the green or just the red, depending on which gene you're talking about. Am I making sense here? Do you guys understand? Okay. No, we're not going to be mixing them together. Right? They are separated. Okay? The green lights are the green lights, and the red lights are the red lights. Okay. All right. So this is the confusing part. Okay? Mutation. Okay, so a mutated proto-oncogene is called an oncogene. If someone has cancer, what kind of doctor do they need to see? What do we call doctors who treat patients with cancer?
Oncologist.
Oncologists, that's right. So oncogene oncologist, they mean the same same thing in the context of what we're talking about. Okay, they have the same prefix, right? So this is actually related to cancer, and I'm going to show you how in a second. Right? So a mutated proto-oncogene is called an oncogene. All right. So what kind of mutation are we talking about here? The kind of mutation is called a gain of function mutation. Causes the growth promoter, growth factor to be permanently activated. Okay, copy it down first, and I will explain. All right, so let's have some visual aids here. Okay, you don't have to copy anything, just like try to listen to what I have to say. So here we have two green lights, okay, because we have two copies of the proto-oncogenes. Proto-oncogenes are the normal ones. They are going to make normal functioning green lights, okay, and this is how normal green light works. This is how normal growth factor works, okay? You turn them on, then the cell sees the green light, they're going to go ahead and divide, okay? But you don't want to turn them on all the time. Sometimes, you turn them off, and then the cells stop dividing. Okay, so divide, don't divide. Right? So that's how it works. Just like real traffic. Okay, green light's on, car goes. Green light's not on, don't go. So that's normal. However, if you have a mutation, okay, if you have a mutation, so the proto-oncogene becomes an oncogene, and that mutation is going to result in a green light that's always on. It's going to result in a growth factor that's always activated. So the situation becomes like this. Okay? The cell divides when they see the signal like this. And then now let's say you're done dividing. You want to tell the cells to not divide anymore. So what do? You turn off the green lights, right? But the other one won't turn off because it's mutated. The other one is an oncogene. It's not going to turn off. Right? So even though you try to tell them to stop, you try to switch off the signal to divide, the other one won't switch off, and your cells will just continue to divide even though you try to turn it off. Okay? So normally it's supposed to be both on, then divide, both off, don't divide. Now we have a mistake in the mutation. It's an oncogene. One of the green lights won't turn off. So even if you try to tell them to stop, they're just going to keep going. And then the cell would divide and divide and divide and divide. That could form like a tumor. Okay? Yeah, tumor, exactly. Does that make sense, guys? Any questions? I'm very nervous about these things, as I can never tell if people truly understand what I'm trying to explain. Okay, I'm gonna explain one more thing before I give you a break. Okay, so over here, just one more thing to add. You saw in my little demonstration, I guess, whatever you want to call it, um, only one of the two copies needs to be mutated for it to cause a problem. Okay, uncontrolled. I'm just saying for it to cause uncontrolled cell division. Okay, we have two copies, but if only just one of the green lights won't shut off, then it's gonna keep on dividing and dividing and dividing. Okay. Do you have a question for me, Fahima? I know it's complicated, but do you have a specific question, certain part that doesn't click? Do you want me to clarify?
No, but it's too hard.
Yeah, sometimes you're not sure what to ask either, right? Like, yeah,
Mm,
yeah. So that's okay. So just take some time, and then, if you have questions later, email me. So what happens when both copy are mutated? You're talking about the green lights here? You're talking about the growth promoter, Cheyenne?
Yes.
Yeah, so like it would just be equally bad. Okay, like one copy is already gonna cause a problem, right? And then like, if another one is broken as well, then you know it's it's even more aggressive growth, in a sense. Okay. You know what? Let's take a break, guys. When we come back, we'll do the red light. All right. 5 minutes, 1:37. See you in a bit. Hey, let's keep going. Now, the green lights are only half of the problems, unfortunately. We also have to take a look at what happens if these tumor suppressor genes um, creates problem. Okay, so let's write them down first, and I'm gonna try to do the demo for you and see if it's gonna help you a little bit more, better. Um, Unlike the proto-oncogene, where when it mutates, it changes name. There are no special name after it gets mutated, okay? But instead of a gain of function mutation, the problem that we have here with the tumor suppressor gene is actually the opposite. It is a loss of function mutation, okay? Think of it this way. Like if you have a red light that's permanently on, it's not going to cause cancer. Right? Cancer is basically uncontrolled cell division, right? So gain of function tumor suppressor gene mutation is not going to be a problem. Uh, Loss of function mutation for proto-oncogene is also not going to cause cancer. If you have a green light that doesn't turn on, that's not going to cause the cell to divide in an uncontrolled manner. Okay, so loss of function mutation for tumor suppressor gene causes the growth inhibitor to not function, or it's permanently turned off. In this case, both copies of the gene, both copies of the tumor suppressor genes need to be mutated for it to cause uncontrolled cell division. Okay. So let's take a look. We have two copies of the tumor suppressor gene producing the growth inhibitor, the red lights, and the red lights are on. Right? So if both red lights are on, does the cell divide or not divide? Not divide. Exactly right. Red light tells them to stop. So no division. That's fine. Okay. Why are they not dividing? Well, who knows? Maybe there are DNA damage that cannot be repaired. Maybe there are telomeres are too short. Maybe there's not enough resources. Whatever the reason, we are not going to let the cell divide. So both of the red lights are going to turn on. Okay. And then if there are no problems, then we turn off the red lights, and then it's gonna be allowed to um to progress, right? Okay, it's gonna allow to go on and divide. Okay, so don't divide. Now, if you have a mutation that causes one of the red lights to not function properly, then it's like this, okay? Like you instead of a oops, sorry. Okay, so like this is go ahead and divide, but let's say you don't want it to divide, right? You don't want the cell to divide, then you gotta turn on the red light. So you try to turn it on, but hey, one of the red light is busted. Okay, one of them has a mutation. It's not turning on. Okay, but that's okay because the other one is still functioning. The cell will see the other red light and know not to divide, right? So normally is like both copy turns on and you don't divide, and then you go ahead and divide. If one of the copy is busted, that's okay. We still have a backup that will tell the cell not to divide. It's only when both copies are broken, when both copies are mutated, you can't even turn on the red light. Okay, you try to turn on the red light and tell them to stop, you can't. Both of the copies are mutated. Then we have uncontrolled cell division. Okay? Uh no, the red and the green are not mixed together, right? So like try not to confuse yourself like that. Just keep them separate. Okay? Are we together? Any other questions about what's going on here with the red light and the green light and all this stuff? Okay. Here is another analogy that might help you understand. Okay, let me just go through all this thing. A gain of function mutation is analogous to like if you're driving a car, if the accelerator is stuck, right, then the car is gonna speed up very, very quickly, right? And that's dangerous, of course. And in the case of the loss of function mutation, sorry, I gotta skip through all these slides. In the case of loss of function mutation, it's like, your handbrakes are not working, but that's okay. You still have your actual brake to stop the car, right? But if both the handbrake and the brake brakes are broken, then there's no way to stop the car, and the car is going to go really, really, really fast, right? So for tumor suppressor mutation, you need two mutations for it to have a problem. For oncogene, just one is enough to cause a problem. Okay. So this is all related to cancer because the mutation results in uncontrolled cell division. And when it keeps on dividing and dividing and dividing, you end up getting a tumor, right? Some forms of cancer are inherited while other forms are acquired. Inherited means it means you inherit genetic codes that make you more likely to have that form of cancer. Whereas acquired means it's something that you get throughout your life, okay? Because of sometimes exposure to carcinogens, right? We talk about UV lights causing cancer. That's why people say to put on sunscreen. But there are many other things that increases your risk of getting cancer, like smoking, drinking alcohol, eating too much red meat, they say, right? So all these things are a contributing factor for acquired form of the cancer. Okay. Now, in reality, it takes more than one mutations to cause cancer, right? Like when I explain it, I made it sound like if one of the green light is busted, then you get cancer, right? That's like an oversimplification, right? It's better to think of your cell as like a city, okay? If the city has one busted traffic light, sure, there's going to be some slowdown and possibly even an accident, okay? But for the most part, the city is okay. But if all the green lights and all the red light in the whole city is malfunctioning, then there are going to be a lot of car crashes, right? Which could shut down the whole city, right? So If multiple mutations happens in the proto-oncogenes and in the tumor suppressor gene, then that's actually what causes the cancer, right? But like I said, I'm trying to oversimplify things here so that it's easier for you to understand. So when someone has an inherited copy of a mutated gene, then their cell already start out with one mutation, okay? And that actually makes it easier for those individuals to get cancer compared to someone who was not born with one mutated copy. Let me try to explain this to you with a diagram, okay? So let's say you have cell here, a bunch of cell. You don't have to draw this. Okay, these are just a bunch of cells. Okay, so let's say one of the tumor suppressor gene got mutated just by chance. Right, it got mutated. Is that gonna be enough to cause cancer? Is it gonna cause, maybe I think it's better to ask, is this gonna cause this cell to divide in an uncontrolled manner? Well, hopefully you're thinking the answer is no, right? Because you still have one functioning red light to stop it from dividing too much, right? So that's okay. Okay, let's say later on in life, the person has another mutation of a tumor suppressor gene in another cell. Is that going to be causing uncontrolled cell division? Okay. Still no. The answer is still no. Okay, look, this one has one malfunctioning red light. This one has one malfunctioning red light, but each of these cells still have one red light that's working, right? So no uncontrolled cell division. It's only if you have two mutations in the same cell, then this cell is going to divide and divide and divide and divide. Make sense? Okay, so what's the chance that you will have the same mutation in the same cell? Remember, you have trillions of cells, okay? So that actually is very, very unlikely. It's like, if you blindfold yourself and then someone spin you around in a room and then you throw eggs at the walls, okay? What's the chance you will hit the same spot twice, right? That it's kind of like that, okay? So very unlikely, my point is. I don't know why that analogy always comes to my mind when I try to explain this. Now, if someone inherits one copy of mutated tumor suppressor gene, okay, from their parents, okay? So all their cells, because it's inherited, all their cells start off with one mutated copy. So what that means is for this person, if another mutation happens in any of these cells, like if it happens here or if it happens here, then these cells are gonna divide in an uncontrolled manner. Okay? So that's why it says here, cancer that are inherited tend to occur earlier in life than cancers of the same type that are not inherited. Okay, let me give you an example. We'll come back to penetrance in a second. BRCA1. BRCA1 stands for breast cancer 1 gene. It is responsible for repairing damaged DNA or destroying cells if DNA cannot be repaired. Okay, in other words, it's gonna stop cell division. So for that reason, BRCA1 is a tumor suppressor gene. Okay, tumor suppressor gene. Right? In the general public, about 12% of women will develop breast cancer sometime during their lives. Okay, 12%. However, for women who inherit a copy of mutated BRCA1 gene, right, if they inherit a broken red light, so to speak, then the chance of getting breast cancer by age 70 is 55 to 65%. Right? And this should make sense to you. Right? It's relatively low for people who don't have one mutated copy because it takes two hits in the same cell for the cell to become uncontrolled. However, these individuals are born with one mistake in every single cell already. So any additional mutation is now going to cause the cancer. Any question about this? Some of you know this actress, Angelina Jolie. She, well, her mother died of breast cancer, and then she did genetic screening for herself, and they found that she has the BRCA1 mutation, right? And so her chance of getting breast cancer is high, and so she didn't want that to be the situation, and she opted to do double mastectomy to remove her breast tissue um, as like a preventive measure, right? So that she's not gonna get the breast cancer. Okay, so if you're interested in learning more about it, you can look it up. Michael asks how fast it is to cause the cancer. Well, again, this is an oversimplification, right? It depends on a lot of things, right? It depends on other risk factors associated with the person. Sometimes it could progress really fast, sometimes not so much. It also depends on the location of the cancer itself as well, right? Even like with breast cancer, it could happen at different parts of the breast, and that have different outcomes as well in some cases. Okay, so here is another thing to be mindful of. There's a concept called penetrance, right? Nowadays, anyone can order like a genetic test, right? They could just swap themselves and send in the sample, and the people would give you a report. The company would give you a report and tell you how likely you are to get certain diseases, and they would even give you your life expectancy, right? Okay, but what those reports often left out is to consider the concept of penetrance. Okay, the genetic makeup of a person is only half the story. Okay, there is a phrase that's called nature versus nurture. Okay? Nature is what you were born with, the genes and stuff like that. But the environment also shapes how those genes are manifested in your life, right? And so just because you inherit a bad copy of a gene does not always necessarily mean it's going to lead to a disease outcome. Right? So penetrance is the concept that describes what proportion of people with a certain mutation will actually end up having the disease or the condition. Right? If 100% of the people have having the mutation ends up having the disease, then it says then this is an example of complete penetrance. Okay, then like for certain you're gonna get it. But often, including cancer, these mutations have incomplete penetrance. Okay, maybe it's like 30% penetrance. Okay, 60% penetrance. Right? Only 60% of the people who inherit the gene ends up getting the disease. Okay, so it's something to be taken with a grain of salt. All right. So what happens when the cell divides in an uncontrolled manner? Well, that will create a tumor, or another name for tumor is called neoplasm. Neoplasm. Okay. So neoplasm or tumor is not necessarily cancerous, right? Oftentimes, the doctor will order a biopsy, which is like a little sampling of the tumor, and then they will do tests on it to determine whether it is harmful or not. So if it's benign, then it's not dangerous. Okay? Usually, surgery will fix the problem. Okay, and there's like no further complication. Okay? Sometimes benign tumors can grow in tricky places, like in the brain, for example, then it's harder to remove. Um, but generally, they are okay. Malignant means it is cancerous. And I'm sure we all know this is something that is progressively going to get worse. And there are treatments available, but not all of them are necessarily effective. So what do they look for to determine whether a tumor is malignant or benign? Well, there are 6 characteristics that they look for. Number one, cancer cells, they have self-sufficiency in growth signal. okay? Meaning they have their own green lights, right? They don't even need other people to tell them to divide. They don't even need an external stimulus to tell them to divide. They will just divide on their own because all their green lights are on, so to speak. And furthermore, they are insensitive to anti-growth signal, okay? You tell them to stop dividing, they won't even listen, right? If the red light turns on, they ignore them, right? They no longer respond to inhibition signals. So together, these two qualities allows the cell to rapidly divide. They can also evade apoptosis. They escape death, right? Normally, when a cell fails G1 or G2, you either tell them to stop dividing or you trigger apoptosis to kill them off, right? But the mutations allow them to evade death. They become immortal. Remember those HeLa cells I taught you in lecture 3? You can grow them in a dish basically forever. They also have sustained angiogenesis. Okay? Genesis is to create. Angio is basically blood vessel. So this is new blood vessel. They create blood vessels that would fuel the growth of the tumor. It takes a lot of resources and energy to grow the tumor. And so that's what the angiogenesis is. Okay, it basically leach away the resources from the body and cause the tumor to grow aggressively. They also have limitless replicative potential, meaning the cell can reset their telomeres so they don't go into growth arrest. They don't become senescent, right? They just keep on going and going. And once they use out all the resources at one location, they start to invade surrounding tissue and sometimes spread to other organs, right? We call this metastasis. So these are the 6 hallmarks of cancers. Okay, some more questions for you to try. We can take it up later, but that's it for lecture 6. Any questions? We have to do a little bit of 7. If you don't have any questions, we're going to move on to 7. Okay. Won't be too long. I'm just going to go through a few key things for you, and then we'll end today. So starting lecture 7, 7, 8, and 9, actually, it's less memorization and more problem solving. All right. So either that's a good thing for you, all right, or if you don't like problem solving, then maybe not so good. But just a little heads up for you. Okay. Lecture 7 is about protein synthesis. We're gonna learn how to decode the ACTGs and use them to build the proteins. Right. But first, a few terms to help you understand what's going on. The genome of an organism is all of its genetic information. If you look at all your DNA, all the ACTGs in your cell, that's called the genome. Okay? The genome is analogous to the whole textbook. Okay? Genome. Now, of course, our genome comprised of 46 chromosomes, okay? 23 from mom, 23 from dad. And on these chromosomes are genes that has the instruction to make proteins for you. So in our textbook analogy, the chapters would be like the chromosome. Okay, there are 46 chapters in the book. And then each chapter, each chromosome would have subsection. Those section would be like a gene. So chapter 1.1 would be like the first gene in chromosome 1. That's gonna be instruction to make protein number 1. Right? Maybe chapter 4.5, that's like chromosome 4, gene number 5, that's gonna make you another protein. Okay? The location of the gene on the chromosome, like where do you find them? That's called the locus of the gene. Right? And that's like the page number. the page number, right? So we actually know the location of all the genes in the human body. You can find it on an online database. We will come back to this in lecture 9. Right? But for now, genome, gene, chromosome, and locus. Earlier today, we talked about homologous chromosomes. These are the chromosome 5 from mom and chromosome 5 from dad, right? 7 and 7. These are homologous. They're same length, same size, and they have genes that are the same type in the same order. But the version may be a little bit different, right? I told you you might got her curly hair gene from mom and straight hair gene from dad. So these various versions of the gene, they are called allele. Allele. So there are different alleles for our genes. Eye color has different alleles, brown eyes, blue eyes, green eyes, and whatnot, right? Before we learn about the central dogma of biology in lecture 3, I believe, right? That's where we take a copy of the DNA and then we make, we take DNA and we make a copy of it in the form of RNA. So that's called transcription. And then to make the protein from it, it's called translation, right? And what I did not tell you at the time is there is a third step called post-transcriptional modification, right? So we're gonna get to that later on in the lecture. This is just like an overview for you. So let's recap quickly why we need to do transcription. First, DNA is too big to leave the nucleus of the cell. Okay, they're not going to fit through the nuclear pores. You can't take the whole bundle out. And even if you could, it's not a good idea because they might degrade in the cytoplasm. So to prevent this problem, you are going to make a copy of the gene that you need to form in the form of messenger RNA. Okay. And also, this allows you to control which gene to turn into protein. Remember, you have the whole recipe book in every single cell, but you're not going to make every single protein that's in the book. Okay. You're just going to make the ones you need when you need them. Right. So you only do transcription for the cell, for the proteins that you want. Base pairing rule, it's something that you should know by now, right? A pairs with T, G pairs with C. The same rule applies to DNA and RNA. A pairs with U because there's no thymine on DNA, only uracil, and G pairs with C. Okay, so these things are old news. Okay, you already know this. So now I'm gonna take a quick note with you, and then that will be it. Okay, it's a short note. It's gonna take like 10 minutes probably. So this is transcription. So this is the DNA, which is double helix. And then it's going to be double helix all the way, but I'm going to just draw a rectangular box here to represent the double helix, okay? Because it's like hard to draw all the way along, and it's messy anyways. Okay, so this whole thing here, this whole box that I've drawn, that represents a gene. And there are many genes on the chromosome, just like there are many sections in a chapter of a book. And just one more time, gene contains instruction. To make protein. To make a protein, usually one gene, one protein, usually. All right. So at the beginning of the gene, there is a special sequence. These are all DNA, okay? These are all like ACTGs. There's a special sequence here called a promoter. sequence promoter. It is an AT-rich region that marks the beginning of a gene. A's and T's, as you know, have only 2 hydrogen bonds compared to G and C, so it's easier to separate it. So the promoter is AT rich so that you can separate the DNA strand easily there. Okay. So step 1 of transcription is called initiation. Initiation. So the enzyme RNA polymerase, because we're building RNA in transcription, RNA polymerase binds to the promoter and unzips the DNA. That's the first step. So I'm going to draw this guy right here. Basically, he's going to move forward, okay? This is the RNA polymerase, not to be confused with the DNA polymerase that we learned last time. Okay, I'm going to zoom in on this middle section so I can draw better. Again, it looks very similar to what we learned last week, but it's not the same, okay? I'm gonna just have some nucleotides here, ATTC. And I'll have the complementary stuff down here. Okay. I'll leave out the 5' and 3', so I'm not gonna, make it too confusing. Now, one of the 2 DNA strands is gonna be called a coding strand. Okay, let me write that down for you. This here, this is the coding strand. You wouldn't know which one is which. I have to tell you on the test, okay? So this is a coding strand, which contains the actual instruction. For making the protein. This is the strand you want a copy of. Now, we talked about this concept before. I'm just gonna reiterate it. You don't have to copy this next part, just listen. Previously, I told you, if I asked you to copy this, then you would just copy it as is. You see ATTCG? In your notebook, you're gonna write down ATTCG, right? No problem. However, for the enzymes that's building these things, right, the RNA polymerase and the DNA polymerase that we talked about last time, if you give it this strand and you tell it to build a strand from it, all it knows how to do is build the complementary strand. Remember we had this discussion? The enzyme only knows to build using base pairing rule. They do not know how to copy directly. That means if you want to have a copy of the coding strand, the enzyme cannot be building from the coding strand. Instead, it is going to be using the template strand. Okay, this is the template strand, which is used by RNA polymerase to create the messenger RNA, which is the copy of the coding strand. So let's take a look here. Okay? Let's say I put the enzyme here, the RNA polymerase, and then it's just moving along. Right? So it will build the stuff. Right? Like, A, U, there's no T, we have to use U instead. Right? And let's say this has been going on for some time, then you're gonna have like a long strand here. So this is the messenger RNA, the copy. Right? It's identical. Identical info as the coding strand. As the I don't know if the color is too confusing. Coding. No, I'm just gonna use the red here. Coding strand. Okay, see the coding strand is ATT, so the RNA is AUU. Right? We don't have T, we have to use U instead. CGA, CGA, CCG, CCG, so on and so forth. It's the same information, right? So we're using the template to build it. So step number 2 is elongation. RNA polymerase unzips DNA ahead and rezipped, I don't know if that's a word, rezips DNA behind. Because you want to keep it the way it was. We don't want to have like a bubble that's expanding like we did with DNA replication. As it builds mRNA using the template strand. Okay, let me go to the PowerPoint quickly to show you something. It's like minting a coin. If you want to make a nickel, 5 cent here, the mold that you use has to be a mirror image of what you want the coin to look like. If you add the metal to this mold, when it comes out, it's gonna look like the way it should. Okay, but the mold is a mirror image. If the mold looks like what you want the coin to look like, and then when it comes out, it's gonna be backwards, right? Do you understand this analogy? In the same way, if we want the coding strand, we cannot build using the coding strand. It's going to come out backwards. It's going to come out as the complementary version. So that's why we're using the template strand to build it. Okay. Finally, last step. Running out of colors here. Okay. This is the terminator. You can probably guess what it is. The promoter marks the beginning of the gene. The terminator marks the end of the gene. So first step is initiation. Second step is elongation, where you actually build a strand. And last step is termination. Okay, so RNA polymerase reaches the terminator of the gene and releases the mRNA. Right? And then it's done. Okay? The DNA is recoil up back to its original state, okay? And then we got the copy. Right? It's very similar to flipping to the pages that you need to copy, photocopy it, and then you close the book, put it back. Okay, you open up the DNA where you need to copy. When you're done, you close it up. That's all. Okay, that's it for today. Very important thing for you to do is to print the slide and lecture notes, and study guide for lecture 7 for next class. Okay, we will be doing some problem solving. Okay, there is a chart that I want you to print as well if possible. I'll post it online afterwards. Okay, it's called the codon chart. But that's it for today. Okay, thank you for coming. See you all on Thursday. Bye for now.